Hello everyone, we are from group 3, myself Vidya Nataraj and my team member Bala Pirami. Today we are going to present paper entitled Spatial Transcriptomics of Healthy and Fibrotic Human Liver at Single Cell Resolution. Our instructor is Dr. Chen Ting Wen. We are from Department of Institute of Bioinformatics and System Biology. This slides gives the basic information about our selected paper. The title is Spatial Transcriptomics of Healthy and Fibrotic Human Liver at Single Cell Resolution. This paper was published in 2024 in Journal of Natural Communications, and the research question is, how are the liver cells spatially organized in healthy and fibrotic liver, and how does fibrosis change gene expression and cell-cell communication? Next, this is the introduction slide. This slide explains why spatial information is important and why sequencing alone is not enough. First, the liver is partially organized into small functional units called lobules. Blood flows from the portal region toward the central vein. Because of this, hepatocytes are exposed to different oxygen and nutrient levels depending on the location. So we have zone 1 near the portrait, which is oxygen rich. Second is the zone 2 intermediate. Third is the zone 3 near the central vein where oxygen is lower. Because of this difference, hepatocytes in different zones express different genes and perform different functions. Now the middle part shows the limitation of conventional single cell sequencing. In NGS based methods like single cell nucleus RNA sequencing, the tissue must be dissociated. After dissociation, cells or nuclei mixed in a tube. NGS can tell us what cell types are present and which genes are active, but it loses the original spatial location of those cells. This is especially important in liver because cell function depends strongly on location. So the solution in this study is to combine two technologies. One is MERFISH and the second is single nucleus RNA sequencing, which is NGS based technology. So MERFISH answers where is the cell, and single nucleus RNA sequencing answers what genes are active. By integrating both, authors connect cell identity, gene expression and spatial location to build a single cell spatial map of the liver. This is the overflow of this work. So the author used two types of human liver samples: healthy liver of 3 donors and fibrotic liver of 3 donors. So from the same samples, they generated two kind of data. On the left side, they used the Murphy spatial transcriptomics. Murphy measured 317 selected genes directly inside intact tissue. So it preserved the tissue structure and spatial location. It analyzed about 90,000 healthy cells and 220,000 cells in fibrotic liver. On the right side, they use the single nucleus RNA sequencing, which is the NGS method in this paper. So single nucleus RNA sequencing provided transcriptomic wide gene expression information and helped identify cell types and disease associated states. They analyzed about 50,000 nuclei in healthy liver and 13,500 nuclei from the fibrotic liver. So then the authors performed computational integration. MERFISH contributed the spatial location and single nucleus RNA sequencing contributed broad gene expression information. So the final output was the single cell spatial map of healthy and fibrotic liver combining cell identity, gene expression and spatial location. Next, this slide explains the NGS method used in the paper, which is single nucleus RNA sequencing. First, the author collected the healthy and fibrotic human liver tissue. Instead of isolating whole cells, they isolated nuclei because nuclei are easier to extract from frozen or complex liver tissue. Next, the nuclei were loaded into the 10x genomics chromium single cell 3 system. In this step, each nucleus receives a unique barcode and each RNA molecule receives a UMI. The barcode tells which nucleus the RNA came from, and the UMI helps count the original RNA molecules accurately. Then the RNA is converted into the cDNA because Illumina sequencing reads DNA and not RNA. This cDNA molecules are prepared as a sequencing library and sequenced using the Illumina Nova sequencing 6000. After sequencing, the data are converted into gene expression matrix where rows are genes and columns are nuclei. Finally, nuclei are clustered and annotated to identify liver cell types such as hepatocyte, macrophages, hepatic stellate cells, endothelial cells, cholangiocytes, and disease-associated states. The reason of single nucleus RNA sequencing was used is that it provides transcriptome-wide gene expression information which MERFISH alone cannot provide. Next, this slides summarize the main computational tools used for single nucleus RNA sequencing data analysis. First, Cell Ranger was used to process the raw sequencing data. It aligned the reads to the human genome and generated the gene expression matrix. Then SUPEX was used to remove the AMP in RNA contamination. Next, doublet finder was used to remove the doublets. After that, Harmony was used for batch correction. Then Surate and SCANPME were used for main single cell analysis steps, including normalization, clustering, marker gene analysis, and cell type annotation. Finally, UMAP was used to visualize the nuclei in 2 dimensions, so liver cell population could be seen as a clusters. So the main purpose of these tools was to convert raw NGS data into clean, reliable, and biologically meaningful liver cell populations. Next, this slide shows the NGS data processing pipeline for single nucleus RNA sequencing. Step 1. The process starts with raw sequencing reads generated from Illumina Nova sequencing 6000. Step 2. Demultiplexing is performed. Step 3. The reads are aligned to the human genome using cell ranges, so we know which gene each read comes from. Step 4. A gene count matrix is created. In this matrix, rows are genes and columns are nuclei. Step 5. Quality control is performed to remove the low quality nuclei, such as nuclei with low gene counts or high mitochondrial percentage. Step 6. Ambient RNA correction is done using SOPEX. This removes background RNA contamination from damaged cells or nuclei. Step 7. Doublets are removed using doublet finder. Step 6. Gene expression values are normalized, so different nuclei can be compared fairly. Step 9. Batch correction is performed using Harmony to reduce technical difference between donors or samples. Step 10. Finally, UMAP visualization is used to reduce the data into 2 dimensions and show nuclei as clusters. Next, the slide step 7. After UMAP visualization, the nuclei are grouped by clustering. This means nuclei with similar gene expression profiles are placed into the same group. Step 12. Then the clusters are given biological names using non-marker genes. For example, marker genes help identify hepatocyte, macrophages, hepatic stellate cells, endothelial cells, cholangiocytes, and immune cells. The final output is an annotated liver cell population. From this, the authors can identify major liver cell types, study gene expression profiles, and detect disease-associated cell states. The key point is that raw single nucleus RNA sequencing rates go through cleaning, normalization, batch correction, clustering, annotation. This converts raw NGS data into meaningful biological information. Next, this slide explains the Murphy's spatial transcriptomics workflow. Murphy's is not NGS, it is an image-based spatial method. Its purpose is to detect selected RNA molecules directly inside liver tissue while preserving the original tissue location. Step 1. Liver tissue is sectioned and mounted on slides. Step 2. The tissue is fixed and stained. Step 3. RNA props are added. Step 4. Fluorescent imaging is performed in multiple rounds to capture RNA signals. Step 5. The fluorescent signals are decoded to identify which gene each signal represents. Step 6. Cell boundaries are detected using nuclear and morphology information. Step 7. The tissue is segmented into individual cells using tools like cell pose. Step 8. The decoded RNA molecules are assigned to their corresponding cells. Step 9. Finally, a spatial cell by gene expression matrix is generated for 317 selected genes. In the study, Murphy mapped about 90,000 cells in healthy liver and 220,000 cells in fibrotic liver. The key point is that Murphy tells us where genes and cells are located inside the liver tissue. Next, this slide shows the output of Murphy. Murphy produces partial gene expression maps. In this study, it mapped 317 selected genes at single cell resolution. So these are the examples of the Murphy outputs, which later my team member will explain about this in detail. Okay. This slide explains why the authors integrated Murphy's and single nucleus RNA sequencing. Murphy's gives spatial location and preserves the tissue architecture, but it measures only 317 selected genes. On the other hand, single nucleus RNA sequencing, which is the NGS method, gives transcriptome-wide gene expression, but it loses the original tissue location because nuclei are isolated from the tissue. So by integrating both, we get the output of cell INT, gene expression, spatial location, healthy and fibrotic comparison, cell-cell communication prediction. So the main limitation of each method is for Murphy's limited to selected genes. For single nucleus RNA sequencing, losses original tissue location.
Thank you, Vidya, for your clear explanation. And now I'm Bala Vidame. I'm going to explain about the major result sections from this research study one by one. So this slide shows the first major result, healthy liver hepatocyte zonation by Murphy. In panel A, the UMAP from Murphy data shows different liver cell clusters. The important point is that hepatocytes are separated into three groups: hepatocyte 1, hepatocyte 2, and hepatocyte 3. In panel F, these three groups are confirmed using marker genes. Hep1 expresses zone 1 markers such as SDS, CYP, 2A6. Hep2 shows intermediate zone markers such as ALDOB and ADH1B. Hep3 expresses zone 3 markers such as CYP2E1 and CYP1A2. In panel H, the clusters are mapped back onto the tissue. This is the key Murphy's advantage. Hep1 is located near the periportal region, Hep3 is near the central region, and Hep2 lies in between them. So the main conclusion is that Murphy successfully shows that healthy human hepatocytes are spatially zonated across the liver lobules. This slide shows the spatial organization of non-parenchymal cells in healthy liver. Non-parenchymal cells are the supporting and immune-related cells of the liver. Here, the authors focused on macrophages, hepatic stellate cells, cholangiocytes, and endothelial cells. In panels A and B, Murphy maps all major liver cell population across tissue sections from different donors. In panels C, E, and G, the authors show where the non-parenchymal cells are located in the tissue. Macrophages, HSCs, cholangiocytes, and endothelial cells show different spatial patterns. In panels D, F, H, and I, marker genes confirm the cell identities. For example, KRT7 marks cholangiocytes, PDG FRB marks hepatic stellate cells, and CD74 marks macrophages. The main conclusion is that these non-parenchymal cells are not randomly distributed. They are spatially organized into specific niches in healthy human liver. This slide shows the NGS-based single nucleus RNA sequence result and how it was integrated with Murphy. On the left, the panel A shows the single nucleus RNA sequence UMAP. Each dot represents one nuclei with similar gene expression profiles clustered together. From this transcriptome-wide NGS data, the authors identify major liver cell types including hepatocyte zones, macrophages, hepatic stellate cells, endothelial cells, cholangiocytes, and immune cells. On the right, panel B is the marker gene heat map. This validates the cell identities because each cell type expresses specific marker genes. The important integration step is that Murphy's spatial labels were combined with single nucleus RNA sequencing molecular profile. Murphy tells where the hepatocyte zones are located, while single nucleus RNA gives broader gene expression information. So the key message is that single nucleus RNA sequence provide transcriptome wide cell identity and Murphy adds spatial context. Together they connect cell identity, gene expression, and liver zonation. This slide shows the zone-specific cell-cell communication. Here the authors used cell phone DB with single nucleus RNA sequencing data to predict receptor-ligand interactions. In the figure, Zone 1, Zone 2, and Zone 3 represent hepatocyte zones. The rows show ligand receptor pairs. The interaction direction can be from hepatocytes to HSCs, macrophages, or endothelial cells, or from those cells back to hepatocytes. The key finding is that different hepatocyte zones show different predicted interactions. For example, Zone 1, Zone 2, and Zone 3 hepatocyte may communicate differently with hepatic stellate cells, macrophages, and endothelial cells. The important point is that single nucleus RNA sequence provides the gene expression depth needed to predict receptor ligand pairs, while Murphy provides the spatial zone context. This slide shows what happens in fibrotic liver based on Murphy's spatial data. In panel A, Murphy identifies different cell states in fibrotic liver. The important hepatocyte groups are portal hep, central hep, fibrotic hep 1, and fibrotic hep 2. In the panel C, portal and central hepatocyte still show some spatial zonation. This means fibrosis does not completely destroy normal liver organization. But in panel D, the two new fibrosis associated hepatocyte states, fibrotic hep 1 and fibrotic hep 2, are spread throughout the tissue. They do not follow normal portal to central zonation pattern. Panel E shows that non-parenchymal cells in fibrosis, especially hepatic stellate cells and macrophages, which also show altered spatial organization. The panel F shows that hepatocyte gene expression changes in fibrosis. For example, fibrotic hep 2 shows increased expression of genes such as SLC7A2, CPS1, ATP9A, POR, and EXT1. So the main conclusion from this results part is fibrosis preserve some normal hepatocyte zonation, but it also creates new injury-associated hepatocyte states with non-zonal spatial distribution. This slide shows the NGS-derived transcriptomic changes in fibrosis. Here, the authors use single nucleus RNA sequencing to study gene expression changes across the whole transcriptome in fibrotic liver. In panel A, the single nucleus RNA sequencing UMAP identifies fibrotic liver cell state, including portal hep, central hep, fibrotic hep1, fibrotic hep2, macrophages, HSCs, endothelial cells, cholangiocytes, and immune cells. In panel C, hepatocyte shows major transcriptome remodeling. Fibrosis changes genes related to metabolism, urea cycle, stress response, and receptor signaling. The important genes include are shown in the are shown here. In panel D, macrophages show changes in immune signaling, cell migration, and cell signaling. In panel E, hepatic stellate cells show changes in extracellular matrix, adhesion, and migration genes. This is important because HSCs are major cells involved in liver fibrosis. So the main point is that NGS-based single nucleus RNA sequence reveals the transcriptome-wide molecular programs deriving fibrosis-associated liver remodeling. This slide summarizes the main conclusions of the paper. First, in healthy liver, hepatocytes are spatially organized from the portal region to the central vein. Second, the authors found that liver zonation is not a strict separation into zone 1, zone 2, and zone 3. Instead, it is a continuous gene expression gradient. And third, Murphy's helped map liver cells directly in their tissue location. It showed spatial patterns of hepatocytes, macrophages, and hepatic stellate cells. Fourth, single nucleus RNA sequencing, which is the NGS method, it provided the transcriptome-wide disease information, and also it revealed metabolic, immune, and extracellular matrix related changes in fibrosis. And the fifth conclusion is about fibrosis created two new injury associated hepatocyte populations. It's called fibrotic hep1 and fibrotic hep2. So these populations expanded in disease and lost clear zonal distribution. So the final message is that combining the Murfish and single nucleus RNA sequencing, it connects the cell identity, gene expression, and spatial location to better understand liver fibrosis in the future studies. And now I'm going to give the answers for that professor asked in our meeting, like during our presentation. So this is about why we selected this paper. So we selected this paper because we are working with liver imaging data, and this study uses spatial transcriptomics to analyze liver tissue while preserving the original tissue structure. And this paper also helps us to understand how NGS based single nucleus RNA sequencing work together with liver imaging data to identify the cell types and transcriptome wide gene expression changes. And the main NGS technology used in this study is about single nucleus RNA sequencing. So it was chosen because Murphys gives spatial information but only for selected genes, while the single nucleus RNA sequencing provides the broader transcriptome wide gene expression from individual nuclei. And the key conclusions from the NGS data is about single nucleus RNA sequencing shows that fibrosis causes major transcriptome changes in hepatocytes, macrophages, and hepatic stellate cells. It identified two fibrosis associated hepatocyte population, and also it shows changes in metabolic genes such as CPS1, GLUL, SLC25A15, PNP, LA3, EGFR, GHR, and LEPR. So the questions after reading is about, after this research paper, do the two fibrotic hepatocyte population appears in all types of liver fibrosis, and also can these cells return to normal after treatment? Could fibrosis related genes be used as biomarkers as therapeutic targets? So it will be useful in the future studies.
So this is our
Presentation.
presentation.
Thank
Thank
you
you
everyone.
everyone.